Hi, hello, and welcome to our Science Cafe tonight, organized by the UMass Chan Postdoc Association at UMass Chan Medical School here in Worcester, Massachusetts. Our event tonight is on the topic of metabolism and disease, and specifically, we will be discussing mitochondria, the powerhouse of our cells. And before we introduce our speakers today, I would like to quickly remind everyone that the views in this presentation and the speakers' presentations are solely of the presenters, not necessarily those of UMass Chan Medical School. Also, our science cafes are recorded and will be uploaded to our YouTube channel here. If you want to share the seminar with your friends and colleagues, also please follow our social media channels listed in the chat box if you would like to stay up to date with our events. Also, if you have a question during the presentation, please type in the chat box and we will ask your question at the end of each presentation. So now we will go ahead and get started. Our first speaker tonight is Levi Ali. Levi holds a bachelor's degree in chemistry from the University of Arizona. He performed cancer research at the Broad Institute for many years. He is now here at the UMass Chan Medical School studying mitochondrial dysfunction in Cohen's lab. In his spare time, he enjoys playing music as a percussionist and making beer. Levi, thank you for being here with us tonight, and the mic is yours.
Okay, so I would like to share a PowerPoint here. So this is the host disabled participant screen sharing.
Oscar, if you want to make him co-host, he would be able to share.
Levi, you should be good now.
Cool, how's it looking?
Great.
All right, well, thank you everyone for attending this Science Cafe. My name is Levi Ali. I'm a PhD student in Cohen's lab at UMass Chan Medical School, and I'll be talking to you a bit about mitochondria and about my research studying them. So I thought I would start things off with something very simple and basic to get people just kind of in the biological world. So I found this video of a person who is going to do a cheek swab and zoom in a bit on cells. So as you can see, she just takes a piece of wood, scrapes her cheek, wipes it on a microscope slide as a little dye, and then she'll put it under the scope, and we take a look, and what do we see in here? 40x, 100x, 250, 500x. And so now you can see here's a happy cell, there's a nucleus, and what's known as the cytoplasm. And within the cytoplasm, there's all of these different, what we call organelles, all these machines that are important for maintaining biological health. And so I'm going to highlight the one I study, which is called mitochondria, so shown in this cartoon here. So what do mitochondria look like? Well, I'm showing you here, this is a single cell under a microscope, and this is a fibroblast, so it's a skin cell. It's not that different from a cheek skin cell. And so in blue is the nucleus, it's dyed, and then all of these things that are green, these are all mitochondria, they're labeled with a green protein. And so you can appreciate there's a lot of them, and they are in different shapes and sizes, and also I should mention that this is a snapshot. So there's a very dynamic environment, and mitochondria are constantly in movement. They're constantly splitting in a process known as fission, or fusing together in a process known as fusion. And so they're a living network. And so if you're like me, you might want to know a little bit more about what they look like besides these rope string looking things. And so what these people did at Northeastern is they took a rat liver and they ground it up and they pulled mitochondria out of that, and then they imaged it with an electron microscope. and they actually do it in what they call a freeze fractionating procedure. So they freeze them and then they hit them, and so they crack open. So you can see here, this is nice and smooth. This is the exterior of a mitochondria, but then this one has been split open, and so when you look inside, you see all of these structures. You also see these spaces, and so I show this to illustrate the complexity. And so this is the world I live in. I try to understand what are these things and how do they work, and so hopefully that's interesting to you. So what do mitochondria do? Well, they're best known for their role in production of energy. Hence, in the next speaker is going to talk a bit more about this. So move on. Mitochondria are known for a role in immunity. If there's, say, a viral infection, this can be sensed by a mitochondria. They'll send signals and react to that infection, so they can help with immunity. Our cells, we have all these ions. One of them is calcium, and so mitochondria actually regulate calcium, which is important for a number of processes. Programmed cell death is a process where sick or dying cells are initiated into a self-destruct mode so they can be cleared and taken out. It's a very interesting and complex process. However, it's initiated within mitochondria. It's important for it to go a certain way. And then finally, stem cells. If people have heard of stem cells, one cell can turn into another, and this is the process known as differentiation. And so mitochondria are actually important in that whole process of cells differentiating. And so I'll also highlight some diseases that are involved with a mitochondrial dysfunction. Some things are related to a small mutation in an RNA in a mitochondria, which might cause a hearing loss or some sort of optical failure or blindness. Or I can highlight heart disease. For example, the heart is a muscle, and so it needs to be pumping all of the time, and that requires a lot of energy. And so you can imagine if there's a problem with energy production, that could be a serious problem. So you see cardiomyopathy and mitochondrial dysfunction. Alzheimer's and Parkinson's disease, we see dysfunctional mitochondria in the brain, and Parkinson's is actually named after Parkin, which is important for mitochondria. And also in cancer, there's interesting metabolism that happens in cancer. And so there's a lot of strange things that happen in cancer with their mitochondria. And I bring all these up just to highlight the importance of studying mitochondria, because we understand how mitochondria function or how they dysfunction or how they can recover from dysfunction. Potentially, we can use that knowledge to come up with new treatments for all of these diseases. So I'm going to highlight one very specific disease that's related to my own research. So this is known as CODAS syndrome. And so CODAS stands for cerebral, ocular, dental, auricular, and skeletal anomalies. I'm showing you here, this is a scan of a person's brain, and you can see there's kind of these white areas and stuff. So that indicates that there's some brain atrophy and there's deformity in the brain. You can see an eye, there's this kind of opaque, milky thing. There's a defect in the vision, dental, auricular, skeletal. All of these different structures are kind of not formed properly. And so all of these together collectively are seen in this CODAS syndrome. And so CODAS syndrome has actually been linked to a very specific mutation in a gene that encodes LONP1. And so if we remember, DNA is this double helix thing, but actually the connection in between these helices is what's known as nucleotides or bases. And so shown here, you have a green and a purple, and here you have a green and a red. And that small change in just one of these bases can actually cause a disease like CODAS if it's in the wrong place, which is unfortunately what happens for some people in these populations. And currently, there's no treatment, and it causes a death in early childhood, unfortunately. So I'll tell you a little bit about LON, the protease that I study, but I also want to highlight this idea known as the endosymbiotic hypothesis. So a long time ago, there was only single cells that existed on Earth, and at some point, one cell engulfed another cell, which, as you can see, is labeled here as the bacterial ancestor of mitochondria. And somehow, that cell inside of another cell managed to survive, and not only that, but it formed a sort of symbiotic relationship, in that it actually together was more functional and able to survive in different environments. And so this is what began what we call eukaryotic life or multicellular life. And so there's properties, and I bring this up to say that there's properties in mitochondria that aren't too dissimilar from things within bacteria. And so when there's properties shared between the evolutionary chain, they're known as conserved. And so LON is a conserved mitochondrial protease. So what does LON do? So it forms a structure here in this cartoon, and it recognizes a substrate. And so what it recognizes are proteins that are damaged or misfolded. And so those things are a problem for the cell, and they need to be dealt with. So what it does is it binds to it, and it uses this magic molecule known as ETP, which Tenzin will tell you a bit about, and it unfolds the protein and feeds it through in what they call the proteolytic core, and that chops it up into little pieces here. And so, it takes a lot of energy and material to make proteins, and so it's important for a cell to be able to take ones that are not quite right and then disassemble them, but keep them in some sort of form that's still reusable. It's a much more efficient process. And so this process of degrading proteins, it's not just in mitochondria, it's in all cells, and there's many proteins that do it, but the one I study is LONP1. And like I said, it's in bacteria, it's in humans, it's in cows, it's in flies, and it's also in this organism that we use in my lab, which is called Caenorhabditis elegans, which is a microscopic soil-dwelling nematode or roundworm. And so I can show you this zoomed-in electron microscope image of a worm, and then I'll show you what I've done with it, which is I've taken one of these worms and I've treated it with a dye known as TMRE. And what that dye does is it actually permeates mitochondria with a healthy membrane potential, which you'll learn more about that in the next talk. But it turns all the mitochondria orange. So you can see here, this is the inside of part of this worm zoomed in, and all of these little orange squiggles are all mitochondria. And so I show you this just to highlight the value. I can't necessarily take a person and look at their mitochondria, but I can take a worm and look at it in real time just by putting a dye on it and putting it under a microscope. So another thing you can do with worms is you can generate mutations. And so what I did is I created a mutation in the gene LONP1. And so I'm showing you here all these little squiggly guys. Those are normal worms with no mutations in LON. And when you introduce a mutation in LON using what is known as CRISPR, which is what I did, now you can kind of see these tiny little squiggles. So this is the only difference between these is a mutation in LONP1. And so that causes these worms to grow very slowly and they lay less eggs. And there's also indicators of mitochondrial stress. So I can show you these worms here. They're kind of green, but not really. But there's a reporter system we have that turns worms green when there's mitochondrial stress. And so I show you this just to illustrate all these different things that you can do with worms. They can take a living organism, you can treat it with something, or you can mutate it, and you can see if that causes mitochondrial stress, which is one of the things we do in my lab. Another thing you can do with worms is called a genome-wide mutagenesis screen. And so what this involves is taking a population of animals. In this cartoon, there's 3, but in reality, it's tens of thousands of them. And you can treat them with a chemical known as ethyl methanosulfate. And what that does is, as we talked before, it can cause these little changes in bases and cause mutations. And so if you mutate a bunch of animals in a plate and they all grow slow, you can kind of see, this is my sketch here. There's small shrimply ones like how we saw before, but in that population, some of them will grow better, and those presumably are mutations that are beneficial. So yeah, they asked me to say something about not all mutations are bad for you. So if you remember the Teenage Mutant Ninja Turtles, I think that was a good mutation. So I have my own Teenage Mutant Ninja worms growing now. So what I did is I took my worms, I treated them with a EMS or ethyl melanosulfate. And so I'm showing you here what we call wild type, or worms that don't have a mutation. The only difference between these and these is that they have the mutation in LONP1, and you can see how much bigger they are as these small guys. However, if I take these worms here, I treat them with a mutation, I select them. Now these are almost the same except for one other mutation, and they grow much better, you can see. So if you remember when I showed you before the TMRE staining, this is mitochondria with healthy membrane potential. I could consider it like a charged battery. So if you look at the LONP1 mutants, Well, now that's very dim, so you could consider that they have like a low battery. They don't have very good membrane potential. They have poor mitochondria. However, if you introduce a mutation from my screen and this mysterious gene known as Y54G9 8.7, now you can see the membrane potential is returned. So something about this mutation helps mitochondria. And so what is this gene? Well, it's a part of a mitochondrial ribosome. So I'll tell you a little bit. You remember we have your DNA, and DNA is read by RNA and taken to this thing called a ribosome, and that makes proteins. And that's in the nucleus and the cytoplasm. However, mitochondria, they have their own DNA, and so they have their own RNA and they have their own ribosomes. And so by mutating part of a mitochondrial ribosome, you can actually presumably correct problems in the mitochondria of a LON deficient worm. So with that, I just want to say thank you to the people in my lab. They've been wonderful and very helpful, and other people in other labs. And I'll wrap up here and take any questions. Thank you.
Great. Thank you, Levi, for a fantastic talk. So we will now take questions. So if you have a question, please type it in the chat box. And maybe I can get us started with a question. So you mentioned that one thing that you can see with the mutant worms is that they are smaller. Do you see any other things happening with the worms, like macroscopically in terms of other things that are sort of wrong with them? And microscopically, do you see a lot of protein aggregation given that the protease function is gone? And if so, is it just a few proteins that are not being degraded, or is it just all mitochondrial proteins that are affected?
Oh, those are both really good questions. So the first one, yeah, there's a lot of things that we see with these worms. I think some of the easiest ones to note is they don't live as long. So there's a test we can do where we just grow worms and just keep them growing and watching them and seeing how long they live. So they have a reduced lifespan. The LON mutant worms are also sensitive to certain perturbations. Certain chemicals will kill them that wouldn't kill a normal worm. So they're very sensitive to certain things that might affect mitochondria or just in general affect health. So in general, they're less healthy and they're less fertile. They don't seem to move as much. They don't seem to eat as much. So there's a lot of problems that these worms have. And then as far as protein aggregation, I've done some experiments to try and look at that, and I haven't seen too much in just a global sense. I don't see a difference, but that might be just due to the biochemistry with the worms. It's kind of hard to look at that, but I am actively checking things. I just got some antibodies recently that should show differences if there's aggregation in those particular proteins. So it's ongoing work.
Okay, great. So we have a question in the chat. For the repressor you identified, do they have homologs in mammalians? And the second part to that question is, if we mutate or knock down the repressor in mammals, are they going to be sick?
Yeah, great question. So yeah, really, as I mentioned before about this, conserved properties of mitochondria. So a ribosome is an extremely conserved, organelle within an organelle. And so most of the proteins in a ribosome are found in a mitochondrial ribosome are found in all other mitochondria, maybe in some different forms, but the ones that I found in my screen, they do exist in a mammalian mitochondrial ribosome as well, although they're not exactly the same. So I'm actively trying to test in a mammalian system these mutations and see what will happen. I can tell you that if you just knock out or inhibit the proteins that I found that will be deleterious to health in a mammalian and in a worm system. And so what's happening in my mutants is it's a bit more sophisticated than just simply a reduction of ribosomes. There's actually altered function instead of just a loss of function. And so I'm actively working on that, but I don't want to speculate too much about what it is, but I have some ideas that I'm currently working on.
So I think a great follow-up question to that is how a ribosome mutant rescues the protease mutant.
Yeah, I mean that's what I'm working on now, and so you can imagine a few things that might be happening when involving this protease. That it's it's involved in degrading a lot of different things. I didn't talk too much about it, but it's also involved in regulating mitochondrial DNA, and so there's an aspect of lawn function that's involved in the production of proteins within mitochondria, and there's an aspect of lawn function that's involved with just degrading general proteins that are imported into mitochondria. And so I'm working on delineating in my mutants, which of these are the actual processes that are affected. And so I can't say any exact explanation of why these ribosome mutations would rescue that. I could only speculate, which I might save for another time when I have a little bit more data, and hopefully people that are interested will follow up with that and attend my public defense talk when that comes up, hopefully in the near future.
Nice. So I have one more question. So have you tried to make more of the mutant proteins or overexpress it either with a plasmid or a viral vector to see if you can reverse the symptoms, the phenotype? Or is that something that's not quite the focus of your work?
So yeah, unfortunately, one thing about the C. elegans system is that overexpression is a bit more challenging. In a cell system, you can kind of just use like a lentiviral transduction or something like that. But in a worm, you actually have to inject into the worm what you'd like to overexpress, and it's usually some sort of array. So it's hard to really control your amount of overexpression. And so we haven't attempted that experiment yet, but I think it's an interesting idea. I would like to try that, but maybe if I can recreate my mutations in a mammalian system, we could try that at some point.
All right, great. Well, thank you so much. If you have any more questions, you can type in the chat box and perhaps we can get to them later. But I think we can now move on to the next speaker. Thank you so much, Levi.
Thank you.
Yeah, thanks, Levi. Now I would like to introduce our second speaker tonight. Our second speaker tonight is Tenzin Ceyang, and she is a research associate in the Spinelli lab here at UMass Chan Medical School. Her research focuses on the flexibility in mammalian electron transport chain that allows cells to function in hypoxic environments. And Tenzin is also applying to graduate school now, and she is interested in studying metabolic diseases. So if you have any tips, you're welcome to talk to Tenzin. So Tenzin, thank you for being here with us, and the floor is yours.
Okay. Hello, everyone. My name's Tenzin Ceyang, and the title of my talk is Reprogramming the Electron Transport Chain. So we can understand metabolism as our body taking last night's delicious pasta and breaking it down into smaller molecules called metabolites, and utilizing those metabolites in different pathways to generate energy and serve other cellular needs. Glucose is the primary source of our energy, and it undergoes a process called glycolysis that produces pyruvate. And glycolysis is just a series of oxidation reactions that release energy in the form of electrons. An oxidation reaction, as a reminder, is when a molecule loses electrons. So here we have an example of succinate, which is a key metabolite in a cycle called the citric acid cycle, losing 2 electrons to form fumarate along with 2 hydrogen atoms. And a reduction reaction is when a molecule gains electrons instead. So here we have the reverse reaction where fumarate gains 2 electrons forming succinate. Now, the electrons that are released during this glycolysis process are deposited onto something called electron carriers, such as NADH. And these carriers help transport those electrons to inside the mitochondria and feed them into something called the electron transport chain that generates energy in the form of something called ATP. So the electron transfer chain, or ETC, is what allows the mitochondria to enjoy its title as the powerhouse of the cell. It's a series of reduction and oxidation reactions in which electrons are fed into and pass through these complexes that are embedded inside the inner membrane of the mitochondria. And we'll begin with NADH, our little carrier buddy, donating electrons from glycolysis to complex 1 and the oxidation of succinate to fumarate via the citric acid cycle, donating electrons to complex 2. These electrons are then moved to complex 3 via the help of an electron carrier called ubiquinone, and ultimately deposited onto oxygen, forming water. Now, as these electrons move through these complexes, hydrogen atoms are pumped across the inner membrane and into this intermembrane space of the mitochondria, where they build something called a proton gradient. And ATP synthase, or complex 5, utilizes this proton gradient and pushes against to generate ATP, which is basically our cell's energy currency. Now, the ATC has many different roles other than just energy production, and I just want to briefly kind of go over them with you to give you a sense of the importance and the impact that this chain has on cellular function. So it supplies electron carriers for other pathways. It can alter gene expression. It makes nucleotides for DNA. It can release reactive oxygen species, which play a huge role in cell death, gene expression, and more. The proton gradient that's built through this proton pumping plays a huge role in regulating mitochondria death, cell death, and heat generation, and of course, energy production. Now, the ETC is actually different depending on the organism. So on the left right here, we have the canonical mammalian ETC. So as discussed in the previous slide, electrons will enter from complex 1 and complex 2, and those electrons will be passed on to an electron carrier called ubiquinone. Ubiquinone carries these electrons to complex 3, where they are then moved to complex 4 and deposited onto oxygen as the final electron acceptor. Now on the right, we have an alternate ETC circuit found in bacteria, C. elegans, and parasitic helminths. And now you can think of this circuit as kind of like a more ancient pathway. And electrons still enter from complex 1 here and are transferred to an electron carrier. But instead of moving electrons to complex 3, this different electron carrier called rhodoquinone donates them to complex 2, which utilizes fumarate as the terminal electron acceptor instead of oxygen. And this also closes the circuit, which completely bypasses complex 3 and complex 4. So a key difference that must be noted here is the direction in which complex 2 activates. So in the canonical mammalian ETC, succinate gets oxidized to fumarate, so that means it loses its electrons, gives it to complex 2, which then transfers it to the carrier ubiquinone. Now in this rhodoquinone-directed electron transfer chain, fumarate gets reduced to succinate, meaning that complex 2 accepts these electrons from the carrier instead of donating. Now, weirdly enough, we see this reversal of complex 2 in mice. And even though that should be kind of impossible if we're utilizing the canonical mammalian ETC. And this kind of begs the question of, could rhodoquinone be present in mammals as well? To test this, we collect tissues from mouse. We perform chemical assays to isolate the potential 2 electron carriers and run them on a machine called an LC-MS. And simply put, an LC-MS is a machine where we run our samples and it separates, identifies, and quantifies small molecules such as ubiquinone and rhodoquinone in a sample. And it turns out that we actually are able to detect rhodoquinone in mammalian tissues. And what's even more interesting is that there seems to be tissue specificity. So you have tissues like the brain and the kidney that have high amounts of rhodoquinone. And then you also have tissues such as the heart and lung, where there's basically no detectable rhodoquinone. And the evolutionary reason for this specificity is interesting to think about. And hopefully, as we go along with this talk, you might start kind of like forming your own hypotheses as well and try to think of like why some tissues have more rhodoquinone than not. And although rhodoquinone can be detected in tissues, we can't detect it in cells. And if we're trying to study the metabolic impact of utilizing a rhodoquinone circuit, we need to actually have it in our cells. So to overcome this obstacle, we overexpress a gene called RQUA that's originally found in bacteria. And what this basically does, it just converts the ubiquinone that cells already have into rhodoquinone. just by changing this one single branch from this methoxy group to an amine group. And now with this tool, we can control how much rhodoquinone and ubiquinone we have in our cells and use it to study its effect on metabolism. And if I haven't harped enough about this already, one last time, a very exciting difference between these two circuits is the utilization of different terminal electron acceptors. So in the ubiquinone-directed electron transport chain, electrons are deposited onto oxygen as the terminal electron acceptor, demonstrating that there's a definite need for oxygen in order for our cells to continue functioning. But in a rhodoquinone-directed electron transport chain, electrons are deposited onto fumarate as the final electron acceptor instead of oxygen, thus also bypassing complex 3 and complex 4. Now, when we grow cells that use a ubiquinone-directed electron transport chain in normal oxygen levels, meaning the oxygen levels we're currently in right now, and then move them to very, very low oxygen levels, also known as hypoxia, we see an incredible amount of cell death. And this is because when oxygen is no longer available to accept electrons, the circuit is put to a stop. Electrons are no longer transferred between complexes, protons are no longer being pumped through the inner membrane, and ATP cannot be generated. Now, what do you think happens when we put these cells that use a rhodoquinone-directed electron transport chain to very, very low oxygen levels, so hypoxia? If we're utilizing a fumarate as the terminal electron acceptor instead of oxygen, we shouldn't be too surprised to see that we see no cell death. And this is because the lack of oxygen doesn't perturb an RQ-directed ETC circuit, because it simply doesn't need it to continue functioning. Now, how does this discovery affect disease prevention and treatment, and why is this important to us? So when you have cardiovascular diseases and are subjected to events like heart attacks, strokes, or embolisms, your tissue is cut off from oxygen for a certain amount of time, and potentially the existence and use of an RQ circuit could protect tissue damage and death that would occur otherwise during these events. There are also many mitochondrial diseases, such as complex 3 and complex 4 deficiencies. Individuals with these mitochondrial diseases don't make it past teenage years on average. So we hypothesize that if these people utilize an RQ circuit, we could potentially help them live longer, since this circuit doesn't require C3 or C4. And lastly, fat cells and cancer cells grow in very, very low, basically no oxygen conditions. And by understanding the mechanisms in which they can grow in these conditions, we can target them for therapy as well. So in conclusion from my talk, mitochondria serve diverse roles in cellular metabolism, primarily due to the ETC. Rhodoquinone is a newly discovered mammalian electron carrier that utilizes fumarate as the terminal electron acceptor instead of oxygen, which makes cells resistant to hypoxia. Thank you.
Yeah. Thank you, Tanzeen. It was really interesting talk, and congratulations to identify a different pathway in mammalian cell. So overall, your study, do you see anytime we can induce the rhodoquinone pathway by switching on or by inducing this pathway? Do you think that you have some kind of inducer, or so that people in the, or you can say the mammalian cell in the oxygen depleted condition can utilize this pathway? Do you have any comment on that?
How we can, could you repeat that question? Sorry.
I mean, because this pathway involved rhodoquinone, so do you think any point of time in oxygen depleted condition we can switch on this pathway other than the ubiquitinone?
I wouldn't be, it hasn't been known yet as of yet, but I really wouldn't be surprised, especially when you have tissues such as like the brain and kidney that have such high contents and amounts of rhodoquinone. Just on like baseline, they have such high rhodoquinone levels. So I wouldn't be surprised if there is some type of like background circuit or backup circuitry to help assist in very like traumatic or like very ischemic incidents.
Yeah, so we have a question in the chat box. Have you tried to overexpress rhodoquinone in mice or in other organisms to see if they are potentially resistant to this drug?
So right now we are working on trying to express RQUA via AAV on mice. So we're still working on like seeing if how that translates in vivo during an ischemic stroke.
Mm-hmm. Yeah, so there's one more question. Do you know if rhodoquinone is expressed in any human cell, either at the protein level or at the RNA level?
As of yet, we do not know.
Mhm. One more question we have for animals living in high altitude area. Is there any change in rhodoquinone expression? Did you study that part?
That would be extremely cool to study, and I wouldn't be surprised to see various levels or various levels of RQ depending on like where you're localized at, right? Where you're living at and what your environment is like. So I would be really interested in seeing that as well, and I would expect that too.
Yeah. So
I have one more question. Can you speculate as to why some tissues in the mice have higher levels of this protein as opposed to other tissues?
So I think it really does have to do with evolution. When you have tissues like the brain, which are so dependent on oxygen, it makes sense for tissues like that to have a lot of rhodoquinone. So I really do think it is kind of like an evolutionary or like need-based. Whether these background or backup circuits are actually being utilized or not, we're not aware of as of yet, but they definitely exist, and they're conserved for a reason. So
one more question. If you compare the ubiquitinone and rhodoquinone, so what are the structure similarity there? Did you study that part?
so structurally, the two electron carriers are within like the same organism are identical, except for the difference between the methoxy group in ubiquinone and the amine group on the side in the rhodoquinone, and that really alters that reduction potential that allows it to deposit electrons onto fumarate.
Yeah, very interesting talk. Thank you very much.
Thank you so
Tenzing.
much.
Yeah. So if we don't have more questions, I think we can wind up. Over to you, Oscar. Thank you, mute.
Hi, thank you so much again for both the speakers, and thank you for all the audience for joining. And finally, we very much appreciate everyone who joined in for today's seminar to help us out by filling out our event survey, which will be found in the chat box. Okay, so thank you all, and see you at our next event.
Thank you, everyone.
Thank you.
Fantastic.
Thank you, everyone.
You may want to stop the recording.
Yeah.
See you later.
See you later.
Congratulations.
Have a good night.
Good
Thank
turnout.
you. Thank you.
Let's wait for one more minute for people to click the survey link. Okay, thank you so much. Have a good night. Bye.
Bye. Good night.
Bye.